translation. We will now take a look at the translation process. This is the process that results in our polypeptide chain, which will then go on to synthesize a functional protein. Translation is responsible for converting the genetic code which was housed in the DNA and translating it into an amino acid sequence. This occurs at the level of the ribosome. This important process uses the transfer or tRNA to read the codons on the RNA strand, mRNA strand. This will help to bring the specific amino acids to the ribosome and to ensure that the correct polypeptide sequence is made. This means the tRNA molecule contains what is termed the anticodon. This anticodon is going to be complementary to the codon on the mRNA molecule. The tRNA molecules will also be attached to the appropriate amino acid. The matching between the codon and the anticodon will occur through the use of hydrogen bonding. Once again, this weak interactive force is very important in ensuring that the proper anticodon and codon are matched together. This, in essence, is how a triplet codon will code for an amino acid. We will now take a look at the function of a ribosome. Note that a ribosome will have a large and small subunit, as we can see in the diagram. The ribosome will lock into the mRNA molecule. The mRNA molecule, remember, is produced from the DNA template, and the mRNA molecule will contain the important codons. In this instance here, this diagram depicts a ribosome that is actively translating a protein. Note that we have 3 different tRNA molecules involved in this process. One of them has lost its amino acid because it's already been added to the polypeptide chain. One contains the polypeptide chain, as we can see in the middle of the ribosome sequence. And also note that we have a tRNA molecule entering into the ribosome with an attached amino acid.
Hello?
Also note that the tRNA molecules
Hello?
contain the
Hello?
anticodon, which is the portion that will lock
Hi.
in to the mRNA molecule
Yes.
using hydrogen bonds.
Where are you? Uh-huh.
This diagram depicts the overall RNA sequence of a typical tRNA molecule.
Hello?
Note that the tRNA molecule will have 2 different ends, a 3' end as well as a 5' end. The 3' end represents the location where the amino acid will be attached.
Cutting out.
Also note at the base of the tRNA molecule we have the presence of the anticodon. This anticodon will once again be complementary to the codon sequence. This means that the anticodon would also have a 3' and a 5' end. Please note the different 2-dimensional and 3-dimensional structures of the tRNA molecule and understand what they represent. When looking at the synthesis of a protein, we will be able to use the codons and the codon chart to indicate what polypeptide chain would result from that particular sequence. For instance, if we have a codon in our mRNA that exists as 5' AUG 3', the corresponding anticodon that would be present on the tRNA molecule would in fact be 3' UAC 5'. This specific tRNA would have the corresponding amino acid, in this case, methionine, attached to it.
Hello?
Note that methionine in the codon chart refers to the codon AUG. Once again, you have to use your mRNA for the codon chart. We cannot use a tRNA.
Okay. I'm going to call you in Messenger, okay?
When a tRNA molecule is loaded up with the corresponding amino acid, we can term this an aminoacyl tRNA. That simply means the tRNA molecule has the amino acid attached to it. There are multiple mRNA codons and multiple tRNA molecules that will code for most amino acids. Remember, there is much redundancy in the code.
You guys are home right now?
Much of this redundancy in the code deals with the fact that there is what we term a wobble. This means that the third position in the base of many codons can change without changing the overall amino acid that it codes for. This allows for some flexibility in the process. Once again, I cannot emphasize enough. the genetic code in the chart that we use is written for codons,
Well, if
not
she's
the
not going there,
anticodons,
then
or
because
not for the
I put
DNA.
money in the US account, I was going to try to get
If
to
you are given
her
a DNA molecule, you must
before
convert
she
it to
left.
mRNA to use this chart. Similarly, if you are given a tRNA molecule, you would have to come up with a
Yeah,
complementary
yeah. I don't
mRNA
know if
sequence in
she
order
was
to
going to
use
go
this
see the
chart.
kids or not.
Please take a moment to reflect on the chart and to ensure that you are able to use this properly. Note that we have the start codon AUG. It would be important to designate the first start codon in your mRNA strand.
Think of.
This will establish the reading frame. You will
probably
then read the other codons in a 3x3
Is
sequence
Renato
and
going?
determine what the corresponding amino acid is.
Oh,
You
okay.
would
Then
continue
maybe I
on
can
this process
e-transfer
until
him
you
the
reach
money and he
the
could just
stop
give it to her.
codons, which we see here in this
Is
chart are
he over
UAA,
yet, or are they
UAG,
leaving from Detroit?
UGA. Though these stop codons do not code for a specific amino
Is
acid,
he
we will
leaving
see
from
what
Detroit?
occurs in the slides following this chart. Also note the position of the wobble. Let's take a look at the bottom of this chart, the left-hand corner, where we have
tomorrow
valine.
morning. Okay, so he can.
Note that valine
Yeah,
can be
he
coded in
transfers
multiple
it. He can
ways.
just take it
As
out
long
of
as you have G
Okay.
and U in the first and
All
second
right.
position,
I'll ask
it
her.
does not matter what your third nucleotide
Okay.
base is.
That was it.
It can
I'm
be
just gonna
U,
see.
C, A,
I
or
don't
G.
need him to, if I'm
In all of
transferring
those
it, I don't need him
instances,
to
you
go
will be
get
coding
anything.
for valine as the amino acid.
Because they
This
close
illustrates
at
the
6,
concept of the
the
wobble.
bank. Okay, so just
Note
keep
that
your phone
in
next to
the
you.
square
I'll
above, we
call our
also have
and
wobble.
see.
However,
I
this
was supposed
is
to do that this
restricted
morning. I
a bit,
transferred it this morning,
meaning that
but I've been running around like a
in
chicken
the
with my head
square
cut off.
above,
And
as
then all
long
of a
as we
sudden I
have
look,
A
I'm like,
and
oh my God,
U,
it's already,
we can code for isoleucine
4:35
if we have
o'clock. So.
UCA in the third position. If we
Okay.
have
All right.
AUG, that designates our start
Did
codon,
Scott talk to
or
Dad about coming to
methionine.
pick me up?
So note that a wobble is still present, however, is
No.
restricted a bit, so that only
Yes.
3
Hello?
different ways and 3 different
Did Scott
codons
talk to you
exist for
about
isoleucine.
picking me up yet?
We're now going to
Picking
take a
me
look
up.
at the structure of a ribosome. Remember, the ribosome
I'm coming
consists
in
of
on
both protein as well as
Wednesday.
ribosomal RNA. In fact, 2/3
Yeah.
of the mass of a ribosome can be attributed to the RNA molecules. Also remember that the ribosome is going to be assembled in the nucleolus. This means that the RNA is going to be transcribed in the nucleolus region from DNA genes. Remember, in eukaryotic cells, this will be done by its own RNA polymerase.
Okay, because it'll be, they're probably going to give us a hassle.
There are 2 subunits to the ribosome, large and small.
Plates on the car.
There are also 3
So we
sites on
plan
the
on doing that
ribosome
when we come back.
that are designated
So he
E for
doesn't want to
exit, P
bring
for
the car
peptide, where the
into the
peptide
US
chain will
because
form, and
they'll
A
give
for
us a
acceptor
hassle,
or
so.
rival region.
Okay.
The
I'm
tRNA
coming around
molecules will
8,
cycle
I think.
through these positions
Around 8
in
of,
growing
no,
the
8 at
polypeptide
night.
chain.
Yeah.
There are 3 stages to the
Okay,
process
so
of
it's
translation:
Wednesday.
initiation,
Wednesday night,
elongation,
yeah.
and
And
termination.
then I'll be there
We
for a
will now
couple
take a
days,
look at those
and then we'll be
specific
driving
steps.
back to be back here for Monday or Tuesday.
The process of initiation begins when
Okay.
the free small subunit of the
Okay,
ribosome
thank you.
is
Love you.
attached to a tRNA
Right.
molecule. The small subunit and tRNA molecule are normally associated with each other within the cell. This tRNA small subunit complex will then bind to the mRNA at the 5' end. This complex will then slide along the mRNA molecule, scanning for the important start codon. Once again, this is usually the first start codon that will be encountered in your mRNA strand. This is very important in designating the overall reading frame for this process.
Hello, hi.
The start codon is easily recognized by the initiator tRNA molecule. This is because the initiator tRNA molecule has the proper anticodon UAC, and it also has the proper amino acid methionine attached to it.
Okay. So
we refer to the initiator tRNA molecule as MET-tRNA.
Tomorrow morning to come over?
The scanning will continue until it reaches the start codon.
Yeah.
An important thing happens at this point. At this point, the large subunit of the ribosome will now attach to the initiator tRNA small subunit complex. When
And
it
he gave it
attaches,
to
the
you.
initial tRNA molecule is now present in the P site of the ribosome. This means that the A site is now open and the E site is also open.
It would be in his, it
This denotes the end
would
of
be in his
the
account. Like I
initiation
can
phase.
e-transfer him and it would be in his account.
The tRNA will then,
And then,
with the correct
I mean, if he
anticodon, will
stops
now
at an
enter into the
ATM, he'd
empty
be
A
able to take it
site.
out, but I don't know if he has
This
time
will
to
lead to
stop
the
at an
formation of a
ATM before he
peptide
comes
bond between
over.
these two amino acids. Once a peptide bond is formed, the initiator
Okay.
tRNA molecule will release the methionine. Please note that the new amino acid is now added to the carboxyl end of the methionine. This means that the N terminus is
Oh,
exposed at the top of the ribosome,
okay.
and the C terminus is where the new amino acids will continuously be added. This will help to grow a growing polypeptide chain in the proper direction from N to C terminus. A translocation event will then occur
Okay.
where the ribosome will slide down the
Because I'd rather
mRNA
give
to
you US
encounter the next
money,
codon. The empty tRNA molecule that has just
right?
dropped
Because it's
off its
worth
amino
more.
acid will now move into the E position. When it does so, it will be removed
Yeah.
from the ribosome. It will then enter into the cytoplasm where it can be free to pick up another amino acid
Okay. Or
to
if
then be used
if he
again.
did leave, if he could stop
The
at an
growing polypeptide
ATM
chain will now
and
be
take
locked
out
into the P
400 for
position.
you, and then I'll
This also
e-transfer
means that
him the
the A
400
position
back.
is now open to accept another
I was supposed to
tRNA
do that today,
loaded up
and I did
molecule.
do it in the
A
morning, and
new
I've just
tRNA
been running
molecule
all
will
over the
enter
place,
into
and
the
then I
A
look at the
site
time,
with
and
the
I'm like, it's already
subsequent
freaking
amino
5 o'clock.
acid.
Um,
These steps will occur over
Otherwise,
and over
Tat
again
can, like I
during
have
the
a US
elongation
account at
phase of
TD,
translation.
it's already there,
This will
so
then
I
stop
can
when a
have
terminator
Tat go and
sequence
get it,
is
but he needs to go by
reached,
6
meaning that a
o'clock,
stop codon
and
in
then
the
you'd have to pick it
mRNA
up
is
from them,
detected.
which
Remember
you're
that there is
running
no
around.
amino
So,
acid that
and I
is
don't want to
present
have Tat
for
to drive, you
that
to drive there
particular
to drop it off
sequence.
to you.
This means
I don't
that
want him
we
driving in
are
like
now
this.
dealing with the action of a release factor. When a stop codon is reached, a release factor will bind to the
Yeah,
location on the
this is like,
mRNA.
I
This
don't,
release
yeah.
factor is important
Okay,
because it can cleave off
call
the
your dad
resultant
and see if
polypeptide
he can
chain
do that. Because yeah, if he can just stop at an
and release it
ATM, he
into
can
the
take out his own
cytoplasm.
money. And then the e-transfer would just go
When this
back and
occurs,
then
the
he can
ribosome
finish that.
subunits will then fall apart
Okay,
and be
so it's
free to
400.
translate another molecule
See
of
if you can do
RNA.
that, okay? And then let me know.
This diagram depicts the elongation process. Note that we are starting at the
Okay,
top of this
and then I'll need
diagram.
his
We
email
have a large
address, whatever he
subunit
wants you,
attached to
whatever
the
he
mRNA
infos his account or
molecule, and we
whatever.
can
Okay,
see that our polypeptide chain is
okay, I love
elongating
you.
in the P position.
Okay, bye.
Also
Okay,
note that
bye.
we have the A position empty for a new tRNA molecule to bind. We will have the appropriate tRNA molecule binding because it will have the proper anticodon sequence. Also note that it is attached to the corresponding amino acid that would correspond to the mRNA codon sequence. Once the new tRNA molecule binds to the A site, we now will have a peptide bond forming between the polypeptide chain and the new amino acid. This requires energy, which we can see denoted in the diagram when we see the conversion of GTP converted to GDP and pi. This is similar to the utilization of ATP molecules. Once the peptide bond is formed, the polypeptide chain will essentially move to the A site. However, because we have a ribosome, this is not the proper conformation for the A site. This will quickly shift and translocate so that the polypeptide will now shift to the P site. As a result, the tRNA molecule that has no amino acid attached to it will now enter into the E site, and it will then be released into the cytoplasm. This then means that a new A site is now empty for the reception of a new tRNA molecule. Once again, this process will occur over and over again during the elongation phase. This slide here denotes the end of the translation process. Here we have a release factor that will come in once a stop codon is recognized. This release factor will be able to cleave off the polypeptide chain, releasing it into the cytosol. It will then cause the dissociation of the large and small subunit. This will then be free to read another mRNA molecule. Also note that this process requires energy once again in the form of GTP. The overall process of translation is a complex process. Once an RNA transcript is created, you can have the formation of polyribosomes that occur when many ribosomes attach on to the same RNA molecule. This can lead to the formation of many proteins at the same time. Once a polypeptide chain is formed, it may require chaperonin proteins in order to take on the proper conformation. There can also be many modifications that need to be made to the protein, such as disulfide bridges, portions of the protein may need to be removed, or phosphate or sugar or lipid groups may need to be added to the protein in order for it to become functional. These are considered to be post-translational modifications because they occur after the protein is translated. Also remember that in eukaryotic cells, we have various types of organelles and the endomembrane system that was deemed very important in producing both plasma membrane proteins or any proteins that need to be secreted or need to be existent in any endomembrane structures such as a lysosome. In order to produce any of these proteins, these ribosomes need to be injected and produced in the rough ER as they are being translated. Remember, this does not need to occur in prokaryotic cells because no endoplasmic reticulum or endomembrane system is present in those cell types. In addition, the process of transcription and translation can occur simultaneously in the cytosol. This diagram depicts the overall process of a specific protein that needs to go through the endomembrane system. In this case, a signal peptide is produced, commonly at the beginning of the polypeptide sequence. This is an important sequence that will denote that we're dealing with some sort of protein that needs to go through the endomembrane system. This signal peptide will be recognized by an SRP or signal recognition protein. Once the signal is recognized and the SRP binds to the polypeptide chain, this will then be directed to the rough endoplasmic reticulum. Once it reaches the rough ER, this will then bind to a receptor protein on the surface of the rough ER. The SRP can now detach, and at this point, the ribosome is locked into the membrane of the rough ER. Translation will then continue. The resultant polypeptide will be injected into the lumen of the ER. This will then travel through the rest of the endomembrane system and receive any post-translational modifications. It will then be delivered finally to its final destination, whether that be the plasma membrane, exterior to the cell to be secreted, or in any of the endomembrane structures such as the lysosome. A mutation results when there is any type of permanent change in the original DNA sequence. However, most of these mutations can be considered silent as long as they do not change the overall structure and function of the final protein. Some of these mutations can be classified as point mutations. These occur when there is a change in only one base pair of the DNA. These can be silent if, once again, they occur in, say, a non-coding region like an intron, or if the codon still codes for the same amino acid. This is what is meant by the flexibility of the wobble position. If the point mutation occurs in the third nucleotide of a codon, it is likely that this will not cause a difference in the amino acid of the polypeptide chain. In this case, this would be silent. A missense mutation, however, exists when the new codon will code for a completely different amino acid. Once again, the severity of this change will depend on a case-by-case basis. If you had an amino acid change in the active site of an enzyme, this could overall result in a non-functional enzyme. However, if the new amino acid is inserted somewhere on the interior of the enzyme, or let's say that it codes for a similar amino acid with similar properties, this could not lead to an overall change in the function of the protein. A nonsense mutation occurs when the mutation creates a stop codon. Once again, the severity of this will depend on where the nonsense mutation occurs. If, say, a nonsense mutation occurs at the beginning of the translation process, this could be very detrimental as the polypeptide chain would be shortened and not be able to take on the proper conformation. However, if this mutation occurs at the end of the polypeptide chain, this may not have an overall effect on the function of the final protein. Finally, there can be insertion or deletion mutations, where the mutation either inserts or deletes a DNA sequence. This can be resultant because of the insertion or deletion of 1, 2, 3, or more nucleotide bases. Many of these mutations are considered to be very detrimental. This is because they can cause an entire frame shift change in the reading of your mRNA strand and thus include a complete frame shift on how that protein is translated. However, if they occur in a multiple of 3, this could potentially be less detrimental. This is because you would simply be losing or inserting a new amino acid, but not changing the overall way that amino acid chain is made. As a result, once again, it will depend on the severity of the mutation will depend on a case-by-case basis. However, in general, insertion or deletions are considered to be the most detrimental type of mutation. Also remember, mutations are considered to be heritable only if they occur in the germ cells. If they occur in somatic cells, they may affect the overall organism. However, they don't have the potential to affect the offspring from that organism.